Hello, everyone. Um, Thank you for attending our virtual conference. I hope that you're enjoying it so far. We will be sending out a survey at the end of the conference for feedback. So if you have any thoughts on how we might improve this or what you liked, what you didn't like, please let us know. We'd really appreciate knowing that information. Today is our last day, and today we'll have the stress biology session and the breeding session, followed by some closing announcements. If you missed any of the presentations so far, most of them have been recorded for the speakers that agreed to allowing us to do that. And we'll be posting them on the conference webpage, so solgenomics.net. Hopefully we'll be able to do that later today. I'm in Eastern Standard Time, so I realize today for me is not today for some of you out there. So our next session is stress biology. So welcome, thank you for joining us. Glenn, Brian, and I will be moderating the session, and we'll have 5 talks. So if you have any questions, please ask those in the Q&A box. We'll try to have as many of those answered as we have time for. But if we do run out of time, there's a discussion session after the stress biology talks. So you can join that session and have a live discussion with the speakers that are able to attend. And we'll post a link for that nearing the end of the stress biology session in the Zoom chat box. I hope you were able to check out some of the posters that we had available on the platform earlier today and last night, my time. We had, I think, about 30 stress biology track posters. So if you haven't seen those yet, they are still available. You might not be able to directly access the speaker for a live chat, but you can still view the posters. So please check those out if you haven't. So with that, I would like to introduce our first speaker, Parvinderdeep Kalan, who is at the Technical University of Munich. And today he'll be telling us about population studies of Solanium2020 that revealed structured major gene-mediated pathogen resistance. So with that, I will activate your mic and you can start sharing your screen. So I think you should be ready to go.
Yeah, perfect. And good. So I hope you see my screen and you can hear me. And greetings from my side. And thank you, Susan, for your kind introduction, and as well as to the organizers to giving me the opportunity to talk in this session. Today I will be sharing my data on part of my PhD project where we looked into a wild tomato species, Solanium2020, and tried to understand major gene-mediated resistance in it. So to do so, we use very well-defined plant pathosystem of Cladosporium fulvum and tomato. Cladosporium fulvum is a biotrophic fungus and has been a major problem as a greenhouse pathogen causing leaf mold. And Cladosporium fulvum has been studied for decades now, and we know very well about the physiology in the domesticated tomato, that it enters via stomata and colonizes the apoplastic space of the plant leaves. And for its successful survival and further multiplication inside the susceptible host, it produces various small proteins or secretes it inside the plant. In turn, a resistance plant can recognize these small proteins, so-called avirulent factors. And this recognition is governed by gene-for-gene interaction. And here, as an example, I'm showing you one of the interaction where AVR9 is a pathogenic protein from Cladosporium fulvum, and CF9 is a receptor protein from the plant. And this, when a pathogen or Cladosporium fulvum in this case secretes this protein inside the plant, this receptor recognizes this. And this recognition further activates a downstream complex signaling cascade with an end goal to give an hypersensitive response. In this, ultimately leading the pathogen to further multiply or grow and making the plant resistant. And these receptors, or wild tomatoes, have been a reservoir of these receptors and have been used in many modern plant breeding programs and has been introgressed inside the domesticated tomatoes and made the plant resistant. And due to this, because the wild tomatoes are reservoirs of these receptors, it is thought that Cladosporium fulvum comes from or originated from the Andes regions. But yet, surprisingly, we don't have a very well documentation of Cladosporium fulvum and its interaction with a wild tomato. And to look into that or to understand that in our group, we use the system of Solanum chilense. I will tell you why in next couple of slides, but before that, I would like to show you a demography of this wild species. You can find this in natural habitats in Peru and Chile. And former studies have shown that these species structure or group in 4 different structures or 4 different regions, central being the ancestral region of this species, and then 2 separate migrational events resulted in 2 separate groups in the southern regions, one being southern highland and one being southern coastal region, and another migrational event also resulted in the 4th group, which is northern. And these habitats or these natural habitats are quite diverse, as you can see in these pictures. When we talk about southern highlands and altitude, you can find these species to be in a rocky, dry climatic regions as well as in the coastal regions. And when we go to the central region of this species natural habitat, it becomes even more diverse. Apart from being present in very dry regions or as well as in the coastal region, it also habitats or grows in lush green mountains. So what this would mean for a pathogen perspective also, that these are different environments. So these different environments are also expected to host or accommodate different kinds of pathogens, and as well as different pathogen pressure depending on the climatic region or depending on the intrinsic climates they face. And so that's why I hope I already convinced you that how diverse or dynamic Solanum chilense habitats are. So now what we did, we took one pathogen, Cladosporium fulvum, and took population from the central region of this habitat and as well as from the southern region, and did infection assays in our lab environment. And here you can see the phenotype that a population from a southern region shows after spray inoculation in the laboratory conditions, it shows disease symptoms, whereas the population from the central region has no visible symptoms. And here it's accompanied with our susceptible control and resistance control showing a lot of hypersensitive response. And this you can see also at with staining and microscoping that you can see the hyphae structure growing inside the plant in our susceptible control as well as in our southern population, whereas we cannot see this growth of hyphae inside the central population. And this we further confirmed with detecting the pathogen load inside the plant via quantitative polymerase chain reaction. And you see that in this graph that the southern population accommodates or have significantly higher amount of Cladosporium fulvum or pathogen load as compared to our central population. So what you can see is or what we can say now from this finding, that Cladosporium fulvum is able to infect Solanum chilense. And we know that Solanum chilense from former studies that it's genetically very diverse. And now we see also that against one single pathogen that it has also differential response depending on the geography of the plant it comes from. So what we can say now that indeed our initial hypothesis that it can serve as good model to study diversity and evolution in Cladosporium fulvum resistance gene, and also we can study the effects thereof in natural populations. But to do so, we cannot just focus on just 2 populations, so we have to take a step further, and we did that, and we took 15 different populations covering all the demography of this wild tomato. And the experimentation, which I just showed you doing around in 150 plants, can be very tedious and time consuming. But fortunately, as I showed you in my introduction, that the interaction of this pathogen with the tomato is governed by gene for gene interaction. So we can exploit or use a very simplistic method where we infiltrate the pathogenic molecules inside the plant and look for the phenotypic response. And here you can see an experiment setup, like how we do so. The 15 populations, which I just talked about, shown over here, covering the north to south. And each population consisted of 18 to 14 plants. And then we infiltrate water as a negative control and to check if there is any wound response after infiltration. And a mix of pathogenic protein, which is produced by Cladosporium fulvum inside the susceptible host. So we infect the susceptible host and then isolate the apoplastic wash fluid from that plant. And that apoplastic wash fluid is consisting of all the secreted proteins by this pathogen. And we see two outcomes after doing that phenotypically. The first outcome being that the plants can recognize this mix of apoplastic, mix of these proteins from the pathogen and gives hypersensitive response. And then there is another phenotype we get that we infiltrate this mix and we see no phenotype at all or no hypersensitive response. And what this means that this plant is unable to recognize the pathogenic molecules, which means that the pathogen can further multiply and grow and makes the host susceptible. And in these plant where it is recognized, you can see there is a hypersensitive response, then the plant pathogen will not be able to further infect the plant and halts the further multiplication and then makes the plant more resistant. And this we can put it in a map. And here you can see that there are single pie over here shows one single population consisting of 8 to 14 plants. And each, the brown region over here represents the proportion of plants which do recognize pathogenic protein. And the green part over here shows the proportion of the plants which do not recognize this protein. And it's quite interesting because this doesn't mean that if we say one accession, I would like to stress over here about this, that we take one individual from one accession and it's a representative of this. No, we do see very diverse responses or very different responses even within a single population. But something which comes really interesting to our eye is that you can see that in the southern group, irrespective if it comes from the coast or if it comes from the mountain region, we see complete loss of recognition. And whereas the central population, some plants do show recognition and some plants do not. And what now we wanted to go two ways. What we want to do is understand why there is a complete loss in the south, and as well as what actually is governing the recognition in our central population. And to do so, we focused on one known key player of Cladosporium fulvum resistance, created as homologs of C. fulvum resistance gene. It is a loci, it's a locus on the tomato genome at chromosome 1 and possesses genes which deliver resistance to Cladosporium fulvum resistance. And the genes varies or changes depending on the species we look into. So far we know from Solanum pimpinellifolium, for instance, that CF9 over here, its gene product recognizes AVR9 from the pathogen, and then it has close homologs 9A, B, D, E of known and unknown functions. And then when we look into Solanum habrochaetus, then it has CF4 gene, which gene product recognizes pathogenic protein AVR4, and then it has close homologs in for A, B, C, and E. And when we talk about domesticated tomato, this region is missing, ultimately making our domesticated tomato susceptible to this pathogen. And now what we can do, we can use the protein of the pathogen or the product of the pathogen and infiltrate in our populations and see what actually is playing the role in our interaction in this. So we did so. The setup was similar, and then we added AVR9 as a pathogenic protein in our experimental setup and infiltration of AVR4 as well. And the populations were northern and central because southern didn't have any recognition. And then you can see, we see actually 4 outcome. I have shown 3 because these are the important ones. The first outcome is over here. We can focus over here, like that the plants can only recognize AVR4, but not AVR9. And there were plants in our screening which were only able to recognize AVR9 and not AVR4. And then more interestingly, which has never been shown before, that the plants can also recognize, there were plants that both of the proteins, AVR4 and as well as AVR9. And the gene product of the recognition, which delivers this recognition of these 2 pathogenic proteins, were earlier thought to be allelic. And this is not the case in our species. And then there was the 4th outcome where the plants were able to recognize only the apoplastic mix, like other proteins, but not AVR9 and AVR4, which would mean that the plants are recognizing another pathogenic protein via another product of another resistance gene. And we can look into the map. And something which comes out over here is that the blue region being the predominantly present in our population, and which is AVR9. So we see AVR9 to be predominantly present in Solanaceae, the recognition of AVR9. But more interestingly, we observe also the gene products of the genes which were allelic, thought to be allelic so far, and the product recognizing AVR9 and AVR4 doesn't seem to be the case in our study. So we see around 6% of the tested plants shows recognition to both. Okay, so now we went further and we wanted to look at gene level, that what actually is happening, what delivers this recognition. Is it just CF4 or CF9, or is it something more interesting happening in this species? And we know about this locus that it's quite diversifying and as well as very prone to many intragenic recombinations. And here you can see in one example, which was shown in 2001 in Solanum pimpinellifolium, the sister wild species of Solanaceae, that the gene, there was an intragenic recombination in some of the wild population of Solanum pimpinellifolium between CF9 and a close homologue 9D, resulting in chimera 9DC. And this gene product recognizes as well AVR9. And this is quite interesting. And you can look, we can look this now in our species also by doing gene-specific polymerase chain reaction, where we just design the primers on the novel region of these genes on 9DC, CF9, and as well as CF4, and understand what is present at genomic level in our populations. And we did this in 9 different populations shown over here. And I can show you one example that a central population over here in the first panel shows the recognition of AVR9. Plus means that the plant recognizes and gives HR, minus means that the plant does not. And interestingly, all the plants show presence at genomic level of CF9. And when we looked into 9DC, we see complete absence. And then we further went to looking into the CF4 region at genomic level. We see some plants show presence and some plants shows absence, but it is not correlated to our response. And then we looked into all the 9 populations, and we can see that irrespective of the response, CF9 is present in all of the plants, and 9DC is absent in all of them, and CF4 is always absent and present, but it's not showing the correlation with our response. But more interestingly, like I would like to focus on one of the plant that it has response to AVR9 and AVR4, and it also has that genomic level, both the regions present. So this would mean that there is much more happening at this locus in Solanum chilense, and it's much more complex what it was thought earlier. And this can be micro recombination or functional chimeras being formed. But the outlook for this part is that we would do in-depth sequencing and try to understand what actually is happening at this locus. And for the last part of my talk, I would like to focus another interesting factor which came out of this study, that we see complete loss of recognition in the south. So what that means and how this can be delivered. And if you remember, I showed you that the recognition of these pathogenic protein is accompanied or accomplished by the plant receptors. And for instance, the apoplastic wash fluid with or the mixture of pathogenic protein we use is consists of around 70 proteins from the pathogen, and all of these receptors are quite novel and different.
2 minutes, Tom.
Yep. Okay, thank you. And these receptors are quite diverse, and depending on the pathogenic protein it recognizes. And it doesn't sound very plausible that the plants can shut down around 70 or more than 70 receptors, rather than the interesting part over here, which is our current hypothesis that it's the co-receptors which are common in all of these receptor recognition that they shut down. And indeed, we do see that in one of the co-receptor, we find up till 18 non-synonymous SNPs and one indel in the population from south, which has no recognition when compared to the population from the north. So yeah, now we would further also look into this with full genome sequencing of more candidates and try to understand what actually is happening at the underlying immune receptor complex. And with this, I would like to conclude my talk and this study that I hope I convinced you and I showed you that Solanum chilense differs in resistance against Cladosporium fulvum, depending on the population and the geographical location it comes from. And interestingly, we see loss of recognition of avirulent factors in the southern population, and we see AVR9 recognition to be predominantly present, but not the only one. And more interestingly, we see that AVR9 and AVR4 recognition is present, which was earlier thought to be delivered by allelic gene products. And two current hypotheses or current working points which we are focusing on is that we would like to now look or understand the complex genomic structure of this loci in Solanum chilense, and also understand the underlying immune receptors. And with that, I would like to thank a lot of people for making this possible. First of all, Ralph and Remco for giving me the opportunity to work in this exciting project, and our collaborator, Dr. Matthew Houston from Wageningen and Professor Aurélien Tellier from TUM, and Mindy, Geshe, and Danila for being actively involved in this project directly, and group of Remco Stam, as well as chair of phytopathology, for giving a great working environment, and as well as SFB and DFG for funding, and my Glasshouse crew for taking care of these amazing plants and maintaining them, Annalise, Berbel, and Sabine. And for you guys for listening and for your attention. Now I would be happy to answer your questions.
Thanks very much, Praveen David, for a very interesting presentation. For me, it's really nice to see some nice new topical work on the CF9 family of genes. So we have a few questions here. So one is from an anonymous attendee saying, do you think these population patterns between southern and northern groups reflect true coevolution of host and pathogen, or rather a migratory pattern of these wild species?
Okay, so yeah, there are this and that. So we don't know pathogen history actually from these regions. So what sounds a bit plausible that it would be more that it's a pathogen loss in the extreme conditions. And for sure, we have trade-off mechanisms because these populations in the southern region requires not only survival to biotic stresses, but also as well as to abiotic stresses. But this further we can say more in detail once we have the pathogen spectrum from these locations. But so far, we can just say that it seems like a factor of the environmental conditions. Yep.
Thanks. Another question is from Giovanni Giuliano is, are these alleles transferable to cultivated tomato by prime editing?
The honest answer would be, I'm not sure. Yeah.
Maybe something to discuss with Giovanni later. And last, another question from Lucio D'Andrea, saying very nice talk. It was wondering if you check the sequence of the PCR amplicons for CF9 and 4 and related to the pathogen, how this pathogen is geographically distributed.
Okay, so sorry.
Did you get all of that question?
Yeah, this question has been asked a couple of times. We did try to look into the sequence diversity of the amplicons as well as of the full genes, which were with the primary pairs which were recommended in the former publications. And this species is insanely crazy with quite a lot of diversity at this locus. And I would say just that we get so much things to make a lot of educated sense of it. So rather than the next step would be going for full genome sequences, we're coupling with in-depth sequences at these regions to really understand the diversity at these locus. Yeah.
Okay. Right. Well, again, thank you very much for a very interesting presentation, Parvinder.
Thank you so much.
Thanks. So we'll now move on to our next presentation in this session, and that's Andrea Schrader from Aachen University in Germany, and she's going to tell us about resilience to salinity in tomato root transcriptome data analysis.
So, I hope you can all hear me now. Okay, so yeah, well, thanks a lot. Thanks a lot for organizing this nice meeting. And I'm also very pleased that I can talk about this very nice project called the Root Project and present some work of my collaborators and us. So here they are. So I would like to thank my partners from the Root Project for providing me the data of their experiment from those partners from Wageningen, Romjana mainly, and also Shinsavetia from Italy, and Christina and Valeria also come from Italy. And Björn is actually also partner of the Root Project, and I'm in the USADA lab working on this Root Project. And Freya Siegler, a PhD student also from the USADA lab, provided me some scripts I could base my work on. And I also listed the other partners of the Root Project from whom I will not present results today. So we are working on the salinity problem, and as you might all know, salinity is really decreasing the arable land surface, and this is really causing problems like loss of production due to salt buildup, to frequency of almost 3 hectares per minute, as was stated by the FAO in the year 2000. That's quite amazing. And especially in coastal and irrigated regions, there is an increase in soil salinity, and it reaches levels that are higher than tolerated by current cultivars. And we wonder, what can we do to keep these areas accessible for food production? Well, one could identify resilient plant varieties and probably use biostimulants, enhancing resilience to salinity of those cultivars being used already. Now, this translates to our objectives. Well, we want to improve the resilience of tomato towards salinity stress, to prepare tomato as a field crop for the increasing surface of brackish soils. And well, if we think about what it is exposed first to the salt stress and salt affected soils, it's the root tissue, right? And therefore we want to optimize the tomato's root system performance in stress conditions. Well, what is well documented is the above soil tissues response to salt stress. And here you will see always in blue the salt stress treatment applied, and I label in this pinkish color the non-salt stress treated plants. And here I have from my setup in Aachen, I have a few plants, and you see the above soil tissues, so the shoot tissue, and you see that, for example, Heinz is way smaller when treated with salt than in the untreated version. And we can see even more for Heinz, Moneymaker, and Moneyberg at the bottom. I plotted a few other phenotypes that I acquired, like the water content, which is reduced upon salt stress, the leaf length, which is shorter upon salt stress, and also the maximum stem diameter, which is reduced upon salt stress. This might be well known, but however, we also need to consider that when we acquire our root phenotypes and our root data, which we want, of course, to set relative to the rich body of available shoot data already in the public. So here's an experiment from our Wageningen partners, which grew some tomato plants on plates for 10 days at different salt concentrations. And we see that the root growth is actually impaired upon salt stress, and the asterisks indicate always significance relative to the control treatment of no salt. And we also see that the number of lateral roots is reduced upon salt treatment. And this is in Microtom and Moneyberg here as an example. Well, in the root project, we will integrate several types of data from phenotyping, from metabolomics, and also from transcriptomics, which I will talk about in the coming slides. And we are using a core collection. Actually, our partners in France are using that in other parts of the project. And we are analyzing also selected varieties, mutants, and overexpressors, and all that is integrated in the end. So, and as I mentioned, one possibility to encounter these problems with these higher salinity is to apply a biostimulant. And which characteristics should such a biostimulant have in our case? So these should be substances which are stimulating processes to enhance abiotic stress tolerance. In our case, salt stress tolerance. In our case, it's applied to the rhizosphere. And we need to check first if our used biostimulant is really acting as a biostimulant and enhancing resilience to salt stress and tomato. And indeed it does. It does. So our Italian partner first tested it on lettuce, and they found that there is an increase of total root mass by 40% when they applied the biostimulant without salt stress treatment. But in tomato, they tested the salt stress treatment then as well. And what we could see here first, there is an increase of root fresh weight up on biostimulant treatment without salt stress. But also there is an increase of fresh weight when there is salt stress, and here's the salt stress control. So we could say that the biostimulant can compromise for the effect of the salt stress on the root fresh weight. So the experiment that was conducted is using 3 replicates of Heinz in each case, and we have treatments of the biostimulant in red and also of salt, which is applied to the plants. And after 2 weeks of treatments and 4 weeks of treatments, so the early and late time point in the following, there's root material taken and harvested. Then I got the data from Novosec, actually sequenced these samples, and I got the data and I could align more than 90% of reads could be aligned. And I used ETAC 4.0 for that. So I could use my transcript per million tables and feed that into a PCA. So here you see a scree plot, and this might suggest, so the first 3 dimensions might be sufficient, so we will see if this is really the case. Let's start with the first 2 ones, explaining most of the variants actually. So let's start now with the PCA plot in the center and then come to the extended version thereafter. You see in pinkish again the no salt stress treatment, and in blue the salt stress treated samples. And we can see that PC1 nicely differentiates the transcript level among the different time points. The time points are in light gray as a contour for the early time point, and in black as a contour for the late time point. And we can also see that PC1 also differentiates for salt stress treatment as indicated by color. So there might be something for the biostimulant in the lower left corner, but we need to have a closer look at this later. So, and then we have PC2, which is a bit more specific. And PC2 is actually, it shows that there's a salt stress dependent suppression of transcript level modification only at the late time point. So in the salt stress conditions, these transcripts seems not to be differentially regulated throughout development. Okay, so what can we do with that? We want to have some informations to have a look on the molecular mechanism and reveal these thereafter. For that, we need to have a bit of more information about the genes underlying this. And therefore, we want to have a look at the functional categories these genes can be sorted in. And this actually we do with the Mercator 4, which assigns genes to MapMan categories or bins. And these are of a specific kind. They are coming in a hierarchical order. And these bins are non-redundant. So this means that I could sum up several sub-bins into a master bin, for example, and have then, when I provide that as a long specific table, I can read again back to these specific sub-bins. And this is actually what I'm doing here with the presentation. So here I'm showing an overrepresentation analysis using these Mercator bins. And we used an enrichment factor of course, of more than 1. So the threshold for the p-value from the Fisher exact test was 0.01. And in white, you see the lowest p-values, and in the darker colors, you have higher p-values. And I will show you on the next slide a few examples for this. Like here, for example, I picked out the secondary metabolism terpenoids. So the highest level bin would be the secondary metabolism, and then the second level would be the terpenoids. So, and of one of those sub-bins in this master bin, there was one of those sub-bins among the top 5 Mercator bins that were overrepresented. And I did that for each PC and also for each direction of the PCs. And I mean, this is quite fitting. We know that upon salt stress, secondary metabolism is changed. And we see that here for PC1 and for PC2. But of course, if the transcripts are changed, we need also to link that to metabolites. And this will be done in the root project as well, because, you know, if the committing enzyme is not regulated by those below the committing enzyme, then probably this might not have an effect on the substances, or if the enzyme is shut off completely, which is activating a specific substance, this might have no effect in function. So there's something other, there's another nice thing about these extended PCA plots. We have the 5 top genes, which are dominating the loadings for each PC again in each direction. And I picked out one example here again for you, which is a potassium cation transporter, which is also well known for salt stress response to be differentially regulated. Actually, this one I think is not published, and we see that without salt stress applied, the transcript levels are increased, and when there is salt stress applied, we have lower transcript levels, and there is a differentiation between late and early samples, so it is higher in the late samples. This is well fitting to this PC here. So, and then I had to dig a bit deeper to eventually find something about the biostimulant. And I found it here in PC5, and you see that, yeah, thank you. Thank you. So you see it here in PC5 that this is nicely discriminating the biostimulant samples from the other ones. And I would like just to highlight one of these bins here. This is the RNA processing pre-mRNA splicing bin. And this little sign in front of it indicates that there were more than one sub-bin in these top 5 aura bins, which is really showing that these processes are overrepresented in these samples. So we will analyze the splicing variants, therefore, and actually I'm just preparing RNA for sending it away. So, and then when we focus only on the salt stress treated samples, these are actually those samples that were of interest in the first PCA plot I showed you. Then we see also an enrichment here in RNA processes like RNA surveillance and transcriptional regulation. And we will zoom into the nature of transcript level regulation upon biostimulant treatment, therefore, in the future. Or in the near future, actually, it's already initiated. So let me summarize the transcript level modification upon salt stress and biostimulant treatment. Well, it's there, you have seen it. So the selected methods for the root project seem to work. So the salt stress can suppress root transcript level changes throughout development, and transport and secondary metabolite related genes are among the targets. Target candidate genes and processes are selected for verification to explain the molecular mechanism of salt stress response and or biostimulant action. We are investigating possible biostimulant-induced modifications of splicing variants, and we are zooming into the nature of transcript abundance regulation in response to salt stress and biostimulants. And with that, I would like to thank you for your attention and also the organizers for making this great meeting possible. Thanks a lot.
Right, thank you so much. That was a very interesting talk. I think we have time for questions, and we have at least one here. So Véronique Lefebvre from Montpellier in France says, very interesting talk. Thanks. Does the salt stress affect the resistance level to pathogens? And what about the interaction between application of biostimulant and disease intensity?
I have to say that I'm not an expert in pathogen response, but I would assume that there might be interaction. I mean, it always depends actually on which biostimulant is applied, yeah, which substances you have there and how that affects probably pathogens. It might also impair pathogen growth, yeah, but also beneficial bacteria might be, of course, also activated by substances. So I think I cannot give a clear answer on that. So we did not work on that. I'm sorry.
Okay, thanks. Maybe something you can discuss later with Véronique and others. And then another question from Monter. I'm not sure what that is. Yeah, what about the actual exposure for 3 months to salt? Have you looked at that?
Sorry, what about the
actual exposure for 3 months to salt?
Exposure for
maybe
I didn't get the word again, but it doesn't. So you mean the constant exposure to salt?
I think so. Yeah, maybe if the person's online, they can ask it themselves. It's a bit of time left.
Yeah. So if you constantly expose to salt, I mean, I'm doing that actually in my setup in Aachen, and well, then the salt stress response is even stronger. But I did not yet apply the biostimulant to these plants. But this is probably done in France.
Okay. There's another comment about the lifetime for tomato, 2 to 3 months. I'm not quite sure what that means. It's more of a comment than a question. Can you?
If the lifetime, maybe if so, yeah, 3 months is fitting quite well. At least I can only talk about my greenhouse experiences, but we also have field trials. Of course, this will depend on the climatic conditions you are growing them in, right? So our field trials, for example, are in Spain. This will be much different than when I would do it in Aachen, I guess, in Germany.
Okay, again, something maybe for the chat room after the session. So once again, I'd like to thank you for a very, very interesting talk, Andrea, and we'll move on to the next presentation. Thank you. So the next presentation is Aymeric Goye from Oregon State. Over to you. If I said your name right. In New Delhi in India, who is going to talk about decoding regulatory networks of diverse coding and non-coding RNAs towards heat stress response. So, Saloni, are you there?
Hi, everyone. Unable to start. Okay, now you can see me, I think. Yeah, I share my screen.
Yep, you're on.
Yeah, okay. So a very good day to all of you. I am working at National Institute of Plant Genome Research, New Delhi, India. This is our beautiful campus, and my laboratory is somewhere hidden behind this first floor window. And I'm going to discuss about the recent results that we have obtained from looking at the different coding and non-coding RNAs towards heat stress response in two varieties that are showing contrasting behavior in tomato. Now, due to global warming, the future climate is predicted to be warmer, and study of the last 150 years have shown that the rate of increase in temperature in the last 50 years is twice that was observed in the earlier 100 years. Moreover, the information from the Intergovernmental Panel on Climate has shown that by the end of this century, it is expected that up to 4 degrees Celsius temperature could be rising in future. Now, this is going to have a dramatic effect on agriculture, and tomato is heat stress sensitive. And so what we did was we wanted to look at the varieties that are now growing in the warmer climatic zones, and this could be important in identifying the molecular hotspots that could be recognized using cultivars that are having a contrasting behavior in heat stress, which could be channelized into making plants heat stress resilient. So for that, what we did was that we started by collecting a few cultivars that are growing in these warm climatic zones. And further, what we did was that we reanalyzed them in cold and heat stress conditions in the field and in controlled conditions, looking at their survival, looking at their physiology, as well as their yield content. This enabled us to identify a sensitive as well as a tolerant variety. The sensitive one was CA4 and the tolerant CLN. Now in nature, plants have acclimated themselves to protect against harsh conditions because of the daily fluctuations that are present. So to mimic such a situation, what we did was we gave heat stress in a way where the plants were first acclimated, and then it was followed by a heat stress. Transcriptomic signatures were obtained from the tolerant as well as the sensitive cultivar. And we were able to identify differentially expressing genes which were coding, that is the mRNA, as well as we had a pipeline to identify long non-coding RNAs. And what we find is that out of the 30,000 odd genes that are present in the ITAG for tomato, nearly 7,000 of them are differentially expressed in response to heat stress. And most of them are shared between the CA4 and CLN, and it's only the 50% of the genes are showing a cultivar specific bias, and there are only 21 genes that are showing inverse correlation. Now, the most important class of proteins are the transcription factors that govern many biological processes. And when we search these heat responsive transcription factors in the two cultivars, we were able to find that there are 402 heat responsive transcription factors, and of them nearly 30 belonged to classes of transcription factors, which belong to the ERF, the HSF, MIBS, the NAC, C3H, and the CO-like. And similarly, another group of transcription factors that were highly downregulated in both the cultivars belong to the BZIP, the BHLX, C2H2, YABI, and the Werkies. But we focused our study on the inversely expressing genes between the two cultivars. And what we find is that surprisingly, it is not the usual heat stress response genes like the HSFs and the HSPs. Rather, the gene ontology analysis highlights that there is overrepresentation of the endopeptidase activity, the transferase activity, and when we compared the NGS data of those inversely expressing genes with our QRT-PCR data, it correlated very well. To look at what these novel regulators are doing in heat stress, we put another screen by analyzing another tolerant and sensitive pair. And going back to our original PCA plot, we picked the Pusa ruby into the sensitive clade and Pusa sadabahar from the tolerant clade. And a comparative qPCR analysis of the inversely expressing genes showed that these 5 genes were consistently behaving similarly in the tolerant and the sensitive varieties. From here, we picked 3 genes for functional analysis. One of them is an acyl transferase, and it is an enzyme that is important to put the acyl sugar moieties in the presence of acyl coenzyme that is present in the Solanaceae trichomes. Notabilis, that is important rate limiting enzyme in ABA biosynthesis pathway, as well as a proteinase inhibitor PIN2. Functionally evaluating the tolerant upregulated genes using WIX showed that there is decreased thermotolerance when the plants were seen or phenotyped 5 days post heat stress, as is evident from the clear drooping of the leaves. These plants actually showed a very high level of ROS as measured by DAB for H2O2 and had an increased cell depth. On the other hand, analysis of the gene PIN2 that was downregulated in the tolerant variety using WIX showed that reducing its levels gives a better performance in the tolerant background, and this phenotype increased tremendously when we repeated this particular experiment using a sensitive background. Our data that these genes are actually involved in thermotolerance was further enhanced by doing gas exchange parameter analysis. And as you can see that for the genes that are upregulated in the tolerant varieties, that is the notabilis and the acyl transferase, there was reduction in the net photosynthesis and water use efficiency when they are downregulated. However, their stomatal conductance and transpiration rate are decreased. The opposite is true for the PIN2 type of proteins. Further, their fundamental role in heat stress response was corroborated when we see that the genes that are ABA responsive and heat responsive, like the HSFs and the HSPs, were downregulated by qPCR in the tolerant up varieties, but were downregulated in the PIN2 proteinase. Next, we also corroborated our data using overexpression assay, which was a transient one. And after agro co-infiltration, we found that the percentage survival of the seedlings was much higher in the constructs that were picked from the ones which are tolerant upregulated. And the decrease in the hypopatile length post heat stress was less in these 2 genes as compared to the vector control plant. Thus, we find that the 2 genes that are upregulated in the tolerant variety, that is the acyl transferase and notabilis, they are acting as positive regulator of heat stress, whereas PIN type 2 proteinase inhibitor is behaving like a negative one. Now, next question that we asked is that is there a natural variation in the promoters of these inversely regulated genes that could be contributing to this thermotolerance that we are seeing in tomato? So we designed experiments in which first we sequenced the promoters of these genes and did a swapping experiment of the promoters in the opposite cultivar background. As far as notabilis is concerned, there was no sequence variation in the promoters between the CL and CA4. And it was clear that when these promoters were hooked to GUS, It showed a clear upregulation in CLN and a downregulation in CA4, pointing to that it is an inherent cultivar-specific differential transcriptional factor pool present in these backgrounds that is regulating this gene. However, when we looked at the other upregulated gene, that is acyltransferase, we find that there was a mutation in the promoter sequence cis element that can probably, that binds to the AP2 or the NFY transcription factors. So does this really translate into the low level in the CA4? So when we put this promoter of CA4 in CLN background, indeed there was a decrease in transcription. But another decrease in the promoter activity of even the CLN promoter in CA4 background showed that in conjunction with the promoter sequence that is different for this particular gene, their transcription factor pool also plays a role in governing their transcription.
2 minutes, Solanaceae.
Sure. Yeah. So our laboratory is also looking at the different microRNAs, and what we have done is that once we corroborated our NGS data with TaqMan-based qPCR for mature microRNAs, we found that there is a overrepresentation of microRNA 169. Now this is a conserved microRNA family, and to our surprise, while there were 14 loci in Arabidopsis and only 4 mature sequences, tomato, which is nearly 5 times the genome size of Arabidopsis, had only 5 loci in MIR-based repository. So we designed a pipeline to fish out new members of microRNA 169 in tomato. And what we found was that indeed there are more number of loci for A, B, D, and in totality, we were able to increase the number from 5 to 18 for the number of loci that are actively making a precursor sequence. And the total mature forms also increased from 5 to 9. Now, these microRNA genes are distributed on 6 chromosomes of tomato, and they are existing as clusters and tandem as well as segmental duplications. One of them is acting, this pair over here is acting as a bicistronic form, which we have shown that is conserved in Solanum pimpinellifolium as well as phenellii. Now these duplicated genes are present in 4 in number, and they are probably important for the expansion of this particular family and for attaining new functions. And does this indeed give any regulatory diversity to the microRNA gene family? We show here by the duplicated pairs that yes, they show differential expression, which could be important in giving it stress responsive or developmental specific regulations. This particular family is indeed highly heat responsive, and its promoter shows many HSCs. We confirmed all the microRNA 169 members as well as the 26 HSFs by 1:1 Y1H assays, and found that there are 5 members that have HSF binding. We confirmed all of them by transient assays. Here is an example, as well as chip assays. Further, the actual targets of microRNA 169 are the NFY transcription factors, and we were able to show that there are other targets other than NFY belonging to other enzymatic and transcription factor family classes. In the tomato, we have been able to show that on heat stress, there is an upregulation of HSFs, which in turn leads to upregulation of the mature microRNA and downregulation of 2 targets, that is 9 and 10, which upon WIGS show that these are indeed involved in thermotolerance and are acting as negative regulators. Further, we also showed that this particular module is also conserved in Arabidopsis, where we can see that plants that are showing an enhanced expression of 169, reduced levels of NFY-A2 survive much better in comparison to plants that are overexpressing the target, or that are plants that are resistant to the microRNA 169 cleavage, or the ones where
Wrap up
the
now, Solana?
yeah, just have one more slide.
Over time, so if you could summarize quickly, please.
Yeah. So our laboratory has used screening, used the contrasting tomato cultivars to decipher the myrinome and transcriptome, where we have found important leads to differentiate, which actually include defense-related and hormone-related genes, as well as microRNA families, which feed into the HSR response that could be feeding back into the loop. So I would like to thank my laboratory members, all of whom have contributed immensely to this particular work, and the grants received from various government agencies, and my collaborators, Dr. Saurabh Raghuvanshi and Dr. Martin Crespi. Thank you.
Okay, thank you very much. A couple of questions have just popped into the box, but again, I think in the interest of time that we've overrun a bit, these will have to be saved for the discussion section after the session. So thanks again, Ronnie. Now we're into the final talk of the session. So this will be given by Ghulam Jalal Ahmed from Henan University of Science and Technology in China, on nitric oxide is involved in the WORK-E81 transcription factor regulated stomatal movement and drought tolerance in Solanum lycopuscum. So over to you, Ghulam.
Yeah, thank you. Can you hear me?
Yep, loud and clear.
Okay, thank you.
So to share your screen.
Okay. Can you see my screen?
Yeah, you're on, so you can start now.
Sorry.
It's all good, I think.
Okay. Is it visible, my screen?
Yes.
Okay, thank you. So thank you very much for giving me opportunity to present my work in Seoul International Online Meeting 2020. My name is Ghulam Jalal Ahmed. I'm an associate professor at Henan University of Science and Technology. Today, I'm going to tell a piece of work which is about WORK-E transcription factor 81, its role in stomatal movement and drought tolerance. So we all know that drought is an important abiotic stress. And with the climate change, the intensity and adversity of drought has increased in the last couple of decades. Every year, we have to experience a huge yield penalty because of drought. If we look at the food emergency percentage in developing countries, we can see that drought contribute the highest compared to the other factors that cause food emergency. So this is very important to understand plant response to drought and to develop technology that can help us to fight against drought stress. In general, plant response to drought mediated by two broad kinds of proteins we call functional proteins, and another group is regulatory proteins. The functional proteins group includes water channels, transporter, detoxification enzymes, earlier proteins, key enzyme for biosynthesis of osmolytes. On the other hand, regulatory proteins that includes proteases, abiosynthesis-related enzymes, protein kinase, phosphatase, and essentially transcription factor. Transcription factor are the key molecular players that regulate numerous plant biological processes. So far, a number of transcription factors have been shown to regulate drought stress response, including Warkey transcription factor. Warkey transcription factor regulates numerous plant biological processes and responses to stresses. Previously, it was thought that Warkey are involved in biotic stress response, but recent studies showed that Warkey are also involved in a number of abiotic stress response, such as salinity, drought, heat stress, cold, heavy metal stress, and so on. However, most of these studies that unveil the role of orchids are conducted model plants such as Arabidopsis and rice. Only few studies were conducted in other plant species. If you look at the left-hand side of the screen, that you see Warkey such as Warkey 41, Warkey 22, and Warkey 33. These have been found in different species to positively regulate water stress response. On the other hand, if you look at the right-hand side of the screen, you see the Warkey like Warkey 17, Warkey 27, and Warkey 33. This Warkey's gene regulate drought stress response negatively. So that means Warkey regulate water stress response both positively and regularly. It depends on the species and the kind of stress. However, the studies on functional characterization on Warkeys in tomato are very limited. We just have a couple of examples, and very recent has been shown on the screen. Several years ago, Huang et al. Did a genome analysis of Warkey transcription factor, and they identified about 81 Warkey transcription factor. And they clustered them into 3 groups, and these are distributed in 12 chromosomes. They also did some expression analysis and found that some of the Warkeys are regulated by biotic stress and abiotic stress. For example, Warkey 81, which are induced by biotic stress as well as the abiotic stress, like salt stress. We did some preliminary screening and look at the Warkey 81 and Warkey 80. And we found that Warkey 81 is a group 3 Warkey, and it is predicted to be nuclear localized. And we developed this tertiary structure. To understand its role in stress response, that means the drought stress response, we silence these genes by virus-induced gene silencing. We also confirmed that a highly heterologous counterpart, Warkey 81, which is Warkey 80, was not affected by silencing Warkey 81. Then we exposed this silence plant and the non-silence plant to the water withholding, that is the drought stress, and found that Warkey 81 expression induced over time and gets peak 8 days after water withholding, then it's declined. If we look at the phenotype of these plants, we see that Warkey silence plants actually shows some sort of tolerant phenotype. That means Warkey silencing enhanced drought tolerance. Then we look at the photochemical efficiency of photosystem 2, and we found that under normal condition, there is no difference at all in the FVFM. However, under drought stress, the Warkey silence plant showed increased FVFM ratio. We also looked at gas exchange parameter, most essentially the net photosynthetic rate. And we found photosynthetic rate drastically decreased under drought, but Warkey silence plant showed increased photosynthetic capacity. On the contrary, we found that stomatal conductance decreased more significantly, and as well as the transpiration decreased more significantly in the Warkey silence plant. which resulted in a significant increase in the water use efficiency of the Worky silence plant. So we also exposed this plant to different water, substrate water content, and found that this actually the water stress actually induced oxidative stress, which was marked by high relative electrolyte leakage, MDA content, and reactive oxygen species accumulation. If we compare with the non-silence plant and the silence plants, we found that silence plants showed less oxidative stress, suggesting that Worky silencing actually mitigated drought-induced oxidative stress. We also overexpressed the Worky81 in Arabidopsis. And when we look at the phenotype, we found that the Worky overexpressed Arabidopsis plants actually showed less tolerance to drought, and which is accompanied by the high levels of electrolyte leakage and lipid peroxidation. Now, we wanted to understand what could be the possible reason. We all know that water deficit normally occurs when the efflux exceeds the influx, where stomata plays an important role. Stomata is a channel that control transpiration stream. We also know that phytohormone ABA and signaling molecule hydrogen peroxide and nitric oxide are involved in stomatal movement regulation, and some transcription factor that activate or repress the target genes, which eventually control these signaling molecules. Therefore, we checked how the stomatal conductance was regulated under drought stress. We found that after exposure of plant to drought stress, the stomatal conductance decreases over time and is consistently decreasing. And the decrease was more significant in Worky silence plant compared to the non-silence plant. When we checked the stomatal density between these two genotypes, we found that there is no significant difference between the non-silence plant and silence plant, suggesting that the reduction in the stomatal conductance is actually not because of the stomatal closure, not because of the stomatal density. When we check the water loss from this Worky silence plant and the non-silence plant, we found that Worky silence plants actually showed less water loss compared to the non-silence plant, suggesting an involvement of the stomata in regulation of water loss. As
2 minutes, Golan.
Okay. As I told that there was no significant difference between the stomatal density, then we check stomatal aperture, and we found that under drought stress, the stomatal aperture of Worky silence plant significantly decreased compared with the non-silence plant. On the other hand, the length by width of the stomata increased in the Worky silence plant compared with the non-silence plant. Then we look at the ABA content in these two genotypes, and we found in the Worky silence plant, the ABA content slightly increased, but not significantly. When we applied exogenous ABA to these plants, I found that the Worky silence plant showed sensitivity to the exogenous ABA, and stomatal conductance decreased more rapidly. Likewise, the stomatal aperture, that is the length by width, also increased more rapidly when we applied exogenous ABA. So that it suggests that the Worky 81 mediated stomatal movement is facilitated by ABA. Then we checked H2O2 content, that is the signaling molecule ROS content in gourd cell. And we found that the H2O2 accumulation significantly increased under drought in the worky silence plant compared with the non-silence plant. We also checked the transcription level of some important stomatal movement pathway genes and found that, most importantly, the RBOH expression was significantly induced in the worky silence plant.
Sorry to interrupt, but we need you to wrap up. But also, we can't see your screen. Can you quickly share it again, because it's disappeared from the window?
Okay, sorry.
But if you could summarize in the next the next minute, would be good. Thanks.
Okay.
Our time for the session.
Okay. Shall I stop sharing, then start again? Do you mean?
You have your screen's disappeared from the window, I think. So if you could just
is it okay now?
Yeah, now it's okay, but could you please summarize in?
Okay, I'm summarizing. So we found that the GARS cell H2O2 content increase in the worky silence plant compared with the non-silence plant. And then we look at the nitric oxide content in this silence and non-silence plant, and also found that under drought stress, and silence plant showed increased nitric oxide accumulation, which is also consistent with the NR activity and NR expression in the worky silence plant. So we checked the transcriptional start sites of the NR and found that there are 3 double box elements. So we thought maybe these are the targets of the worky A21. To summarize my story, that we found the worky A21 is a negative regulator of drought tolerance, and we think that worky A21 actually suppress H2O2, nitric oxide, and ABA content, which eventually inhibits stomatal closure. So since multiple signaling molecules such as ABA, H2O2, and nitric oxide have been found to mediate stomatal closure, so deciphering the crosstalk and the functional hierarchy of the worky A21 regulated stomatal movement could be an exciting topic for future research. So finally, I would like to thank my collaborator, Dr. Yuan Chang and Dr. Xin Li, and I'd like to acknowledge the funding from National Science Foundation and Henan University of Science and Technology. Thank you.
Okay, thank you very much. There are a couple of questions in the question and answer window, but I think, again, we're a good 5 or 6 minutes over time for the session. So if Chandni and Jonathan and anyone else who wants to ask questions of any of the speakers, if you could join me in the chat room, or whatever they call it, straight after this, that'd be great for the next 20 minutes or so. I'd like to thank all of the speakers this afternoon for a very interesting session on Solanaceae stress biology. So thank you, everyone, and we'll close the session there.